Okay, so this is, actually I just got into the country late yesterday. I was in Chile for the last 4 or 5 days. So Steve and I have not had an opportunity to dry run this, so we're on the fly. But he's a good organizer of slides, so I think this should go pretty good. The topic that we will be talking about today is probably the #1 problem as far as infectious diseases in the swine industry at the moment. It's an emerging disease that we've never had. I'm not discounting PRRS, by the way, but if you happen to get PEDV, you will be well aware of it. We're going to be talking about Porcine Epidemic Diarrhea virus. It's a new emerging disease, and it is in the U.S. Right here? Okay. So, the disease, you'll see vomiting and occasional diarrhea in sows and gilts. Very, very severe diarrhea in nursing pigs. And actually, if you're a nursing pig and you get it, you're dead. Long and short of it. Very, very, very nasty. You'll see some pictures here that will leave an impact. Diarrhea and occasional vomiting in growing pigs. The older you are, the less severe the disease is, but they maintain it for a while and shed it for long periods of time. Clinical PEDV is indistinguishable from TGE. How many have old-timers remember epidemic TGE? Yeah, absolutely devastating disease. You do not want it. This looks exactly like it. They're both coronaviruses, but there's no cross-protection between these two different coronaviruses, nor serological distinction in a diagnostic assay. PEDV is a swine disease only, so pigs that get it and recover, i.e. grow, finish, etc., the meat is perfectly fine, no association with human disease at all. PED has been in Europe for a long time. It was first recognized back in 1971. It's moved since then from the European continent to Asia. The traditional vaccines that were developed against the original European strains really don't work against the strains that are out there right now. Incubation, once the pig's exposed, 36 hours or less, 24 to 36, depending upon the age of the pig. They're going to break with disease. The literature says, usually quit shedding about 11 days post-exposure. Well, wrong. We can show you that it's out to at least 28 days. A colleague of ours at Iowa State, Darren Madsen, he's got data that go at least 35 to 42 days. And on the farms, cyclic through a farm, it lasts for a long, long time. Laboratory diagnosis is really important in these older pigs because a lot of times they're going to present and the clinical disease that you see really isn't all that apparent, post-weaning diarrhea. And if you're older than that, you might not even see any clinical disease. Okay?
So we're going to go to the barns now. And before I start that, I have to say one of the proudest things I've ever been associated with Kansas State, my alma mater, was talking Dr. Hesse into coming to K-State. He's put us on the map. So this is what happens when you go to the barns. Dr. Matt Ackerman's a colleague in Indiana. He was one of the first areas in which this disease broke out. One of our blessings is that the two big outbreaks that occurred simultaneously in Indiana and Oklahoma were being covered by two of the very best swine veterinarians in the country. They're both bulldogs. They wouldn't give up till they got an answer. If it had been stupid old me, it had been 3 weeks later before we'd have known it. What you got is these little babies with diarrhea. That obviously was not the...
Here, you want to use this?
No, I just want to click the right button.
Okay. I did that too.
Lots of scours, lots of diarrhea. Dr. Hesse's point about diagnosis is right because at this point you don't know if it's TGE. You don't know if it's bad rotavirus. You don't know if it's hot E. coli. In the nurseries, what you get, if we can knock the lights down a little in the front, please, is just a bunch of skanky-looking pigs that have got post-weaning diarrhea, and we're all used to seeing them. It could come from a number of things. Plenty of that, we know it, but that's what really happens.
Okay, so I was talking to Matt when he gave this picture out, and I said, wow, this is really devastating. That really leaves the impact. And he goes, Dick, that's day 2 of the outbreak. At day 3 of the outbreak, we had 3 pickup loads full of baby pigs. This is a devastating disease.
So it's bad, and it's one that we've got to understand. So this is for all the students in here. Dr. Hesse, what's going on?
All right. So, as we said just a little bit ago, PEDV is caused by a coronavirus. That's what coronaviruses look for. Probably the most important end as far as, since I'm a vaccinologist for 27 years, I'm most interested in this portion right here, because that's where we're going to develop neutralizing antibodies. It's a single-stranded RNA virus, and it infects the villi, predominantly infects the villi. Actually, all we've been able to show is infection of the GI tract, and takes normal-looking villi that look like that and reduces them to what you see there. It results in malabsorptive diarrhea, and the pigs die from dehydration. That's not cool. Mortality, young pigs, virtually 100%. This is a pretty darn hot strain. Actually, it's probably hotter. The variation that we have is probably hotter than the original strains that broke in Europe. Older animals, it's more or less age-dependent as far as the severity of the disease that you see. However, shedding of the virus lasts for a long time. Once again, it's a pig-only disease, does not affect humans. It's important. We don't want any scare of the safety of our food. I think we already talked about that. The virus that we have in the US is most closely related to viruses that are circulating in China. These viruses, so basically the flow showed up in the Asian countries, Korea, Japan, most recently in China, and the virus is continuing to evolve. The spike protein changes are such that the old vaccines no longer are effective. I don't know if it's that much hotter. I mean, Korea 10, 15 years ago had pretty severe disease from PED. But when I was over in China last year, got to visit a number of different labs, and I said, what disease do you have that we don't want to get? Every single lab said PEDV. It's a seasonal disease. If and when it shows up, it is absolutely devastating. We know that. Now,
one point about this for everybody here, we use the word epidemic when it's people. So if influenza comes sweeping through, it's an epidemic. In animals, it's epizootics. So a little confusion there, but that's part of the veterinary world, is make longer words and more confusing world.
Okay. So one thing you might notice that this says Veterinary Diagnostic Lab, Iowa State University. I have used a lot of slides from fellow colleagues from the different diagnostic labs and the professional organizations across the industry. I want to point that out, that the camaraderie, the unification of the different labs to combat this disease, the sharing has been just absolutely amazing.
In 40 years, nothing has come close to all the universities pulling together, sharing stuff. National Pork Board was on board. Everybody I know, except the government, came on board and did a good job. I'm so unsurprised.
Now, Steve. Okay, you shouldn't be saying that. All right, so we did not have this disease officially until the 17th of May, I think it was. Yep, in 2013. We don't have enough time to actually go through all the history and the transmission and the places that it was that we didn't know it was, but we'll just kind of give a brief overview. I was confirmed by the National Veterinary Service Labs on the 17th of May. That initiated a really coordinated effort, mainly by the American Association of Swine Veterinarians and the National Pork Board. And they both stepped up. They put their money where their mouth is. They were extremely active in trying to understand this disease, trying to get control of it, and dollars. The Pork Board has put up close to $1 million right now just to combat this disease. That's, I think, really, really important. The diagnostic labs, as I said earlier, they really teamed up and shared knowledge, resources, observation. Can't say enough good things about that. Makes me proud to be a diagnostician with these guys. Trends? Up, up, up. Sow finish or boars. How's it spread? We'll talk a little bit about this. Steve will talk more. Trucks? Nasty. Why are trucks so nasty? Because of the volume of virus, the amount of virus in feces. We're talking between 8 and 10 logs of virus in a gram of feces. Well, infected pigs can poop a lot, and that's a lot of virus that's out there.
And
you walk, go ahead.
Kent Schwartz, Iowa State University, pointed out that 1 thimbleful of diarrhea from a 3-day-old pig contains enough virus to infect every pig in the U.S. This is a fecal oral disease. You'll hear that so much, but it's manure.
Yep. Walk through a truck, walk back and forth into the process, you'll talk more about that, and you just cross-contaminated everything. Okay. Some evidence of aerosol transmission. That's not necessarily exactly what the laboratory data say, but in the field we're seeing it. So lots of variability. So we can talk about that later. That's hypothetical at this point in time. Bottom line is, once it's into an area, it gets around, and when it does, it's not good. Breeding herds. That's where you see the most of it. Oklahoma, Texas, Southeast, North Carolina. Bad news. All those pigs moved to the central U.S. All right, so this is the frequency of cases, new cases reported. All right, so this is when it first broke. Had our big peak back in June or so. Kind of leveled off over the summer. This is the most current data as of, 2 weeks ago. It's going up and up and up. This disease is a seasonal disease. We're in the season. Now, actually, we're not even there yet. I fully expect it to get a lot worse. As from the trend line that we're seeing, we're well on our way. How bad is it going to get? I don't know.
So this is where the pigs are in the U.S. Lots of them in Iowa. Here we are in Kansas, North Carolina. We're an industry on wheels. We're moving about 500,000 pigs a week into Iowa alone, and we move about 1 million pigs on rubber every day. Well, anytime you're moving pigs, you're moving manure, and this is a fecal oral disease. Okay, so now I need that one because I just blew my battery. Here's where the cases are as of 2 weeks ago on the 11th. And one of these is the right button. Okay. Here we go. This just tells us how many cases have gone to the diagnostic labs from the various areas. And you'll notice Kansas is up in the upper areas. Almost all of that's right down here in the corner because our friends down at the Seaboard and Texas and Hitch and all of that crowd have just been smoked. It's been awful. And it's re-breaking now. So a lot of cases here, a lot of cases here. Iowa's getting bad. Our friend Ian Levis, talking about Iowa Select last week, said, yeah, in the last 8 to 10 days we broke 16 sow farms in our company alone. So you get some idea about how fast and how hard this thing's moving. So what do we got to do to figure it out?
All right. So samples to submit to the diagnostic lab. All right. So intestine, fresh intestine, and fixed. A way you can look at the histopathology. And if you want to try to isolate virus from intestinal content, fresh intestinal content is much better than feces. But if you want to just diagnose it by PCR, feces, fecal swabs, oral fluids are absolutely great for that. Virtually the same observational data that you get with the PCRs with any of those samples. Same way with intestine. We now have some antibody assays that are coming online. There's a number of them, additional ones that will be coming online, but at K-State we're doing IFA. As a vaccinologist and virologist, I want to know antibody status at every step along the way. It's a good sample to pull for a variety of reasons. This is a slide, and the main reason I wanted to show this is how the disease actually works. Immunohistochemistry, Darren Manson is responsible for generating this data. Darren Manson at Iowa State. These are the villi. This is normal villi in a neonatal pig, a nursing pig. Look how long and big they are. One thing I didn't realize, and Darren taught me, newborn pigs are born with mature enterocytes. They mature in utero. I didn't really realize that. Okay, and so that's why they are so extremely susceptible to PED infection. The virus loves mature enterocytes. That's why older pigs only have mature enterocytes at the tips of the villi, whereas newborn pigs, it's the entire villi. And when they become infected, it's a massive, overwhelming infection. This is that same structure, intestinal structure, 36 hours later. Gone. All the villi. Their absorptive capacity just no longer exists.
And there's a real important take-home message for all of us out in the field, too, with this whole process, because brown is virus. Antibodies stain the virus, so these cells are full of virus. That's 8 hours after the pig got infected. Over here, 36 hours later, there's very little virus there. Now we're going to have to go in the field, collect virus to expose sows and gilts so they become immune. Guess what? We got to move really, really fast. You've got to gather material that's going to have a lot of virus in it, which means that you can't do it 3 or 4 days later. This is a high-speed response situation, and this kind of shows why that becomes a necessary step.
Quite literally, a day late and $1 short for feedback. Okay, so the Pork Board was very, very generous in providing initial research funds to conduct work on PED. I was fortunate enough to submit a proposal and got funding for that. We wanted to look at where the virus was camped out in the pigs, how long it was going to be shed. Do you establish a carrier state? What's the antibody response following infection? And is there aerosol transmission? So those are the things that we wanted to address. We were looking at 4-week-old feeder pigs, and these are the preliminary results. They're getting more and more solid every day, but as I learn more and more, my interpretation is changing. So that's kind of good. Lots and lots of people involved in this work. Most of the people over at Mosier Hall in the diagnostic end of things. The molecular group, by the way, they did an absolutely heroic job of turning these samples. I mean, we were flooding them with lots and lots of PCRs. They just, boom, did it all. Totally impressive. Bob, Maureen, you guys, Megan, yep. They were in the BRI collecting all these samples. Drew, I don't know if you're here either, but yeah, lots and lots of work going on. And Yang, one of our new faculty members, is actually working on a pretty neat ELISA assay that's going on. So once again, lots of people working on this. The study itself was done at the Biosecurity Research Institute, the BRI. This is, for all practical purposes, a foreign animal disease. It's not a notifiable foreign animal disease, which kind of is an interesting thing. Steve might have a comment or two about that, but anyhow, at least the initial work we chose to do, or it was decided to be done, let's put it that way, in the BRI. So what we did, because we were interested in aerosol transmission, we divided the room in half. One half is dirty, the other half is clean. Dirty means infected with contact controls. Clean means just pigs in the room breathing the same airspace but not inoculated. So you can see, and this is the dividing line, and this is just so a sense of perspective. The plastic sheet there is to prevent water droplet when the pens are being cleaned. But there's open airspace here, and then the whole front of the room is open. Okay, the garb that you wear, I mean, this is a BSL-3 ag facility. We did it as it was BSL-3 ag. otherwise known as an incredible amount of work. So that's kind of the setup. BRI. The study, because of the pig space available, it's not a real huge study. Only 33 pigs, and we divided them into 3 experimental groups. The inoculated ones, they got the virus via their intranasal inoculation and then orally. Contact controls, these 2 groups were commingled together, and then the aerosol control group that was just sharing the same airspace. The challenge, this is actual feedback material that we got from Dr. Ackerman, one of the first vets that was supplying all the initial information. And it was actually used in a sow herd to immunize them, or a controlled infection, let's just say that. It had a CT titer. CT refers to real-time PCR, so a nucleic acid titer of 22. Actually, when we tried to isolate virus from this, we were unable to. So bottom line is fecal material that's been frozen and thawed is not good for virus isolation. Lots and lots of infectious virus. Pigs are much more sensitive than cell cultures. And once again, 4-week-old pigs. Reason we went with 4-week-old pigs is we wanted to have live pigs that we could study and monitor. The Taj Mahal for pig rooms. You're not going to get much cleaner than that. And oral fluid sampling. More pigs make for more efficient oral fluid sampling, and it's a good method. What did we see? Well, the clinical disease that we saw was not all that severe. We were a little bit disappointed. Actually, I was expecting that we were going to have sick pigs within 24, 36 hours. Well, they didn't really get sick. They didn't really get diarrhea. Finally, we had one that vomited, and I go, ah. In the meantime, I was on the phone with Darren Madsen at Iowa State saying, Darren, they're not getting sick. What's going on? He said, just wait, they will. Sure enough, they did. We did have, all the pigs became infected that were inoculated. All the contact controls became infected. They were all shedding. The degree of diarrhea that we saw was not the really, really severe diarrhea. It was the cow pie type diarrhea. For the most part, it was not as severe as what I thought it might be. We thought we would probably have some die. It wasn't quite that bad. Shedding, what did we see? So we looked at fecal shedding. We looked at nasal swabs, the virus in the nasal swab. Now, originally I was calling this shedding in the nasal swabs. I am thinking that over again. The data that we have says maybe not. So what do we see? Inoculated here, no virus at 24 hours. I thought that was a little strange, even in the nares, because we loaded them up with a lot of stuff. The contact controls and the inoculated animals showed virtually the same profiles as far as virus load. So this is real-time PCR. The lower the CT, the greater the amount of virus. So it's kind of reverse of what you normally think. This is the negative line, and basically, yep, fecal shedding was actually about 10 CTs. That's 1,000 times more shedding, or greater detection at the nasal swab, or fecal swab versus nasal swab. And we had some interesting hits in the aerosol nasal. Aerosol fecal, they're flat negative. Okay, I think, so I'm a graph guy. I had Steve put that slide back in so I could speak off that. So I think I'm saying all this in the bullet points that we're just observing. Basically 24 hours, 48 hours is when we first saw a detectable virus. Fecal shedding and nasal was greatest, no, that didn't go back, did it? Yeah, 5 to 6 days post-challenge. 28 days. We could track shedding up to 28 days in some of these animals. Oral fluids, same thing. 28 days. That's a valuable tool for screening a lot of animals, oral fluids. And the data that we saw with oral fluids is virtually identical to the data that we saw with fecal swabs. So that's a nice tool to have. I'm talking too much. We had those interesting hits that we had with the aerosol in the nares of the aerosol control pigs. Well, we wondered about that, so when we took environmental samples in the room itself, guess what? Viral nucleic acid everywhere. Okay. The aerosol control pigs did not exhibit clinical disease. They did not exhibit fecal shedding. They had it in their nares, but then they also had it in their pens, in the food trays, in the V-troughs, but no productive infection. That's kind of contrary to what the field's saying. Because there were questionable results, we actually sequenced the PCR product of that, and yes, indeed, that was positive for PED nucleic acid. It's real. Lab gossip. Okay, so experimental data and real-time data that you see in the field. Our data says, no, you probably don't see respiratory transmission of PED. I mean, it's pretty solid. We collected tissues. We looked at PCR. Well, we looked at immunohistochemistry in the turbinates, in the trachea, in the lungs, in the bronchial lymph nodes, in the spleen. We could not find virus anywhere. That says it probably doesn't occur. Well, it's not what the field data says. It looks pretty real that it does occur out in the field. So we need to understand why that. Is that because of the bioassay that's being used? Is that because of the age of the pig? Is that because of UV sunlight killing the virus? Is that because we had so much disinfectant in the room that it was unbelievable? There's a lot of things we don't know. Other things from the field. Water transmission. You run PCRs on the sewage ponds. Guess what? You're going to find viral nucleic acid. Some of the more interesting ones is they capture some geese and swab the outside of the geese, positive for viral nucleic acid. Now, bioassays need to be done on all these samples to really get a feel for what's what. We just don't know what the most sensitive system is. Everyone's doing the work in weaned pigs. Well, it probably should be done in nursing pigs. Immunohistochemistry, yep, we infected them. Yeah, we didn't put that in the right spot. But, okay, so the lesions, histological lesions were minimal. IHC was there. We looked at all these other tissues, and we can't find it except in the GI tract. And we had nice, clear-cut, working immunohistochemistry. So we know that our results are pretty accurate. The other thing I think that's really important is antibody response following inoculation. So everything's seronegative at the time of inoculation. Nice, vigorous antibody response at 14 days post-inoculation. This happens to be an indirect fluorescent antibody test. Ying's group is actually doing the same type of work with antigen ELISA, looking at the nucleoprotein, and her data looks virtually identical to this as far as the profiles. So that's useful if you're in a quarantine situation and you want to go two periods of when an expected seroconversion time would be. Just wait a month. And whether, if it didn't happen by then, it's not going to happen. What else? Titers higher than expected. Once again, no evidence of seroconversion in the aerosol control group. I really do believe our data, even though it's in direct conflict with apparent data from the field. We need to investigate this. ELISA, I just talked about that. Some of the other things that are currently underway is multiplexing, which will be kind of a really neat tool. The poultry industry flock profiles a lot. Well, we can start, and we are starting to do some of these serological surveillance tools with Luminex, and then obviously serum neutralization. That correlates best with protection. All right, so once again, the common good and sharing, all the diagnostic labs working together. One of the most important goals of our project that was funded by the Pork Board is to develop diagnostic samples that we're sharing with other laboratories So actually 4 different labs, diagnostic labs, have received the complete set of samples that we generated. I know, Barb, where are you? You're sick of making aliquots. So we've actually distributed. The most recent one is the National Veterinary Service Labs. We sent the samples to Sabrina. She's going to be sending them on to Canada as well. So we've got over 1,200 samples that we've distributed throughout the diagnostic world. Oral fluids, I mean, there's a lot, so, and we continue to do that. From my personal perspective, I need to thank these people. Matt for sending us the samples, sharing the information. Ken Schwartz, Darren Manson from Iowa State providing initial reagents and blow-by-blow events of challenge. Darren's group was actually a week ahead of us, so it was kind of useful there. Sabrina Swenson from the National Veterinary Service Lab, she provided the initial virus that we used to develop other assays. Eric Nelson and I and Joe was in on the phone conversations of how difficult this virus is to isolate. We can actually isolate it pretty easy. Serial propagation, that just doesn't work very well. Steve Higgs from the BRI, they stepped up and actually provided the room space free of charge. That's $33,000 worth of room space just to run this experiment. I mean, we are really, really appreciative of that. And then last but not least for us, the National Pork Board for stepping up. It's yours now.
The thing Dick left out about that was when this thing broke the 1st of May in the whole U.S., nobody had an assay to say it was PED. All these labs made the tests by the middle of May. K-State among them leading the charge.
Actually,
technology is pretty cool.
That's kind of an important point, and I'll brag about our molecular group a little bit there. Several of them had some gel-type assays or cyber-green assays, but we actually had, by one day, the first real-time assay, and Jemfa and his group hopefully he's here. Yeah, it was kind of cool. So, but every lab stepped up and did amazing things.
And if you're old like me, that's the first time I've ever seen them all do it together, which was kind of cool. There's big gaps in the knowledge. We still don't understand a lot on transmission. The age differences and susceptibility, we know kind of why the little bitty babies are the ones that really get smoked. We don't have good methods to grow it. We still have to have serology methods because taking blood samples after the fact lets you know which pigs that are now normal did have it before. And develop an efficacious vaccine. I sure hope that works, but there's some awful smart people in Asia that can't make it work, so I'm a little bit worried that that may be a while coming.
Everyone's working on it, but we have a ways to go.
Okay, so it's arrived. Now let's just take another look at it from where I look at the world. And first of all, we need to get a little perspective. When these bad things happen, you know, I've lived through the tail end of hog cholera, and we went through pseudorabies, and we've gone through PRRS, and then we had to have circovirus and APP. We got another one. If I live and practice another 5 years, there won't be any foreign animal diseases left because they'll all be here. Every time Jim needs a program, we have to have a disaster. But let's just stop and take a deep breath a minute. If PED does its very worst, and kills 5 weeks of pigs, kills 10% of the weaned pigs in the U.S. It's horrible. That's $440 million. I want you to remember that number, okay? Because that's important. It's bad, but it has to be held in perspective. One of the challenges for me and for all other veterinarians is that when we go to investigate these things, it isn't just PED that we're worried about when pigs have diarrhea. We've still got TGE. This is an electron micrograph of some viruses. This happens to be the PED coronavirus.
So the picture, actually, I got it.
This is Dick's picture. I stole it.
So I took this picture back in 1981 when I was working at the Diagnostic Lab at South Dakota doing diagnostic EMs. This happens to be TGE and rotavirus, but PED would look exactly like that, but yeah, 81, so.
So we can't just say, okay, there's one disease. Now we got to become multitasking. Never let a serious crisis go to waste. And what I mean by that, it's an opportunity to do things you think you could not do before. This quote has been given to many people through history. This just happens to be the most recent. The mayor of Chicago mentioned it. And this crisis is making us do stuff we didn't think was possible. And I'm proud of this industry for jumping up. The key to this whole thing, it's really about the feces of the species. This is a fecal oral disease. Packing house information and coal markets. The first thing that popped up on that was that some people were thinking ahead and said, let's go see if we might be moving this thing around in slaughter truck contamination and biosecurity. So thanks to quite a few veterinarians, we know that it happens with dysentery and TGE. What about PED? So the middle of June, right in the early part of the outbreak, veterinarians sampled 7 different slaughter plants. Did 100 trailers at each plant, and we do what's called the Swiffer test. You know what the Swiffer is. Well, I hear there's a thing called a Swiffer. But you can take this Swiffer and put some phosphate buffered solution on it and swab the back of a truck and wring it out and have a nice sample. So they did 100 trucks. And they did them using the Swiffer pads. And in the middle of June, before we really knew we had a horrible thing going, 17% of the trucks were already PED positive on arrival at the slaughter plant. The jolt was that 11% of the trailers that arrived negative on their swab test were positive when they drove away. And when that result came out, it was kind of an oh crap moment, because it's like all over but the shouting, because it's going to move. So that brought to light a whole lot more things about, well, how are we contaminating trailers? And the first rule is that all truck drivers enter the plant. Well, we should know this, but we don't ever think about that part of the world. They have to carry the bills of lading into the scale. They got to offload the pigs and pen them in the holding area. Then they got to break the load down into multiple lots, take them to the scale. If there's a dead pig, got to drag him off, get him out of the flow. If there's a downer, you got to go back to the plant, get the sled, go get the pig, go get the downer, take the dirty sled back. It's a minimum of 5 trips into and out of a plant if you're a driver. Now, all of the plants have really stepped up. They've got working groups going on, new rules coming out. They're trying to figure out ways that are more efficient to get a piece of paper from the tractor to a scale across campus, then have the guy carry it in and carry it back. The electronic era, it may be possible, I don't know. But there's changes happening there. Paul Yeske presented last week. He was one of the ones that stayed up all night and swabbed trucks. Currently, trucks that enter a slaughter plant are not only highly likely to be contaminated with PED, TGE, dysentery, and PERS, but washing out the back compartment is likely a waste of time, because a lot of people were washing the back compartment and hosing down the run-up. Since the truck really should be contaminated all the way through because the person's been in and out of it so many times. The next step in this whole thing is, okay, what about people and vehicle traffic? Think about all the movement we have on farms. Wean pigs and culls, maintenance and repair personnel, feed delivery supplies, showers, benches, barriers, workers, and yeah, even veterinarians. Not the most sterile bunch of people. You know, we go where sick things are. Think about that. And survival is enhanced by cold and wet weather. If you look out the door, PED is celebrating today. At freezing, it's virtually indestructible. So to prevent introduction, it's about not moving in feces. Farms are doing a lot of things. We now have on some farms fumigation chambers that everything goes in, from tools, supplies, equipment goes in fumigation chambers. Some have UV lights, fog them with disinfectant before they enter the farms. It's all about protecting home plate. That's where the sows live, because that's where the babies are going to die. We can make the rest of them live, but the ones that only have enterocytes are going to die. Manure spreading and vehicle and all of that sort of stuff that goes with it, I really give a lot of credit to Pork Board, because they stepped right up. And if you go to the website, all of these are out there. Pumping protocols for PED control, for the commercial haulers, for pork producers, for landowners. If you look at the big breaks in Iowa right now, it's still speculative, but what happens in Iowa this time of year is corn gone, bean gone, manure on. And I mean, it's just a traffic run every which way, and sometimes there's a little spillage. And you can track stuff around pretty fast. There's a lot of resources out there. but manure and manure spreading in our present system is probably a big deal. Mortality disposal, that's another one. What should we do? Should we do composting, bury them, render them? These are 3 compost systems in Kansas. We had a chance a couple weeks ago to go visit KDHE, and all the field staff were there in Topeka. My hat's off to them. They are with us 100%, and they feel for us. They're there to help us. They got to go to southwest Kansas and see what it looked like with a rendering system. When you have baby pigs dying at that rate in the middle of June, you got to use dumpsters and rendering trucks. It's not pretty. And it's also because their charge is to be handling the biosecurity and the environmental issues of Kansas. They're to protect the waters of Kansas. And I would suggest that a track hoe and a big hole in the ground, probably not a very good public image, is either for the pig industry or for protecting the environment of Kansas. So I'm a big proponent that I can grind a heck of a big pile of straw pretty fast. They've agreed, Terry Medley and his group have agreed that if we need to have emergency approval for compost sites, write us a letter. We'll try to help you get it to do. So I'm proud of them. Next thing, disinfectants. Okay, we got to have tools. What are we going to do with it? Well, everybody's got a lot of disinfectants. We just don't use enough of them. Real quickly, there's all these different kinds of disinfectants: aldehydes, biguanides, bleach, iodine, oxidizing phenols. It has to do with whether they work in organic matter or don't. The big bottom take home from this is the ones that really work are the really nasty ones. The ones that make your eyes hurt, burn you, stink up the cab of the pickup. They happen to be the ones that work. I believe in hydrofoamers because you get a blanket of foam down, which improves contact time. Disinfectant doesn't kill it the minute you pull the trigger. It's got to have contact time. Foam helps with that a lot.
Okay, I'll make a quick comment about feces. All right, remember Steve was talking about a thimbleful filling a room, every pig in the U.S. So disinfectants. Your disinfectant is 99.999% effective. That sounds pretty good, doesn't it? Sounds real good. Well, the virus is there in 1, 2, 3, 4, 5, 6, 7, 8 logs for sure, maybe 9 or 10. Well, 5 logs versus, let's just say 8 logs. That's 1,000 infective virus units that are still left behind. Disinfect and probably do it again.
It's partly why it has to be cleaned first. Pretty hard to disinfect poop. The gold standard that we're doing in trying to protect home plates are what are called TAD systems, thermally assisted drying and decontamination systems. This happens to be the one from J6. All this is a big broom that you take a power washed and disinfected semi-trailer into, close the doors, turn on the grain dryers, and when you get her to 160 for 10 minutes, virus is gone. And that's become very much a necessity, especially in the breeding stock arena, because we're not going to power wash every trailer perfectly 100% of the time, and the disinfectant's not going to get every place 100% of the time, and some days it's going to be cold and some days it's going to be frozen. TAD units are being built all over. It's changing the culture of how we're going to move pigs in a sanitary fashion. What does it take? Well, Daryl Holtkamp, Roger Main. Roger took his KSU PhD and really keeps doing wonderful things with it. They just took aluminum trays, made up a slurry of manure with PED virus on them, put it on the trays, let it dry, and then they'd take those trays. Now, they didn't disinfect them or power wash them. They just either cooked them, here's 160 degrees for 10 minutes, and then after they'd done the treatment, they scraped the trays off, dosed these baby pigs, and if they got sick, that's what's called, we talked about having, what did you say that was? A bioassay.
Yeah, bioassays.
A bioassay is where you take the target animal and you give them something. So like Dick mentioned, the goose in the lagoon. So if we want to do a bioassay test, first of all, we have to shoot the goose. Second, we have to feed the goose to the 3-day-old pigs. Now you got a bioassay.
But they were probably using weaned pigs there.
They're probably using weaned pigs on this, which aren't near as susceptible. But if you see that, at 160 degrees, 10 minutes, none out of 4, But 145, 1 out of 4 were still positive, 130. If room temperature, 24 hours, 1 out of 4. If they just let it sit room temperature and dry for a week, the virus died. Not very practical parking every trailer for a week at room temperature.
I would also interject at this point as a virologist, if you change some of the conditions a little bit and coat it in protein like serum or something, and then dry it down, it can last for months. We've done that same experiment with FIP and other viruses. And actually, if you think about freeze drying, how you stabilize vaccines, modified live vaccines, very similar process. So how you do it, what the matrix is in the sample that you're drying down and test over time will really affect stability of the agent. So this is good information, but it's not the end-all be-all.
So we're trying to do our very best to protect what we can protect. It's impossible with the technology and what we have to do to move the number of pigs we have to move to slaughter 400,000 a day. We cannot do it with the resources we got. But we got to back up and say, I'm going to try my very best to protect home plate. Well, that's where the sows are, because that's where the babies are going to die. So that's where baking, disinfecting, cleaning come in. Okay, so enough of that. That was all of what I could have, should have, would have done. What happens? I call up, my farm's got it. It's got acute PED or PERS, but pigs are scouring, things are bad. What are we going to do next? And what are the goals? The goals are really simple. There's only 3 of them. Contain the infection, don't spread it. That's the first rule. Don't make a bad situation worse. Second, work with the goal to wean negative pigs. And then just keep them negative the rest of their lives. That's all we're going to do. Thank you. So how are you going to do that? That's the problem. Well, what happens when my herd's infected? The first step we've talked about a lot is the diagnosis is critical. And the critical first step is call. Don't panic. Sit tight. We don't want to miss other agents. We don't want it to be a PERS plus a PED outbreak or a TGE confused outbreak. We've got to get that sorted out. Do not pack up a whole bunch of pigs and feces and carry them back to the office to get ready to mail them home where everybody's going to track through. I mean, don't move it. We'll figure out a way. Our K-State Diagnostic Lab, by the way, has raised the steel curtain of biosecurity to protect. They've done a wonderful job with it. We're going to have to get these samples, get the right samples, and we're going to try to help people get the right kits so we can get what we need to do to get the answer quickly, because we got to move quickly. The second thing we have to do is we have to get that sow herd and the gilt herd all exposed and make all of them ill. Because unless they become ill and recover from the virus, they're not going to be able to protect the future generations of baby pigs. That means a closed herd, so there have to be enough gilts in there to expose too, because Dick already mentioned, they're going to shed virus for quite a while. We can't very well do the whole sow herd and 30 days later go get a load of gilts and come in and watch them go nuts, because now guess what? Here goes the virus again. The details are critical because we don't want an endemic result. Nursery and growing pigs are going to be sick too. And we downplay that a little bit in that we say, yeah, they survive. Well, survival is one thing. Our job is to take care of pigs. We really don't want to have to take a week to 2 weeks of growth off of them if just keeping them warm, dry, comfortable, some electrolytes might have saved them. So we have to be ready to respond for those pigs too. Here's how it goes. It's really easy. You get an acute sow herd infection. What are you going to do? First intervention is intentionally infect the whole population with the pathogen. We got to do it now. So we're going to have to get in and collect manure and tissues from those baby pigs that are just getting sick. It can't be the ones that have been scoured out and are finally dying 4 days later because they won't have much virus. Once you've done this, whether this is PERS or PED, there's only 2 pathways. One's success. We succeed, and sometime later we're weaning negative pigs. The other ones we fail. Well, what is failure? Failure is when you get an endemic infection. And what's an endemic infection? Well, that's one that just keeps on giving. And so I know if I look over here and ask Leo Schwartz what chronic endemic TGE looked like in the nursery 30 years ago, he can tell you. What happens is now every wean group of pigs sits there and scours for 2 or 3 weeks, and we got, you know those gains that you guys have on your charts?
Yeah.
You don't have negative numbers on yours. I can show you negative numbers. Average daily gain, minus. That's what happens. We don't want to go here. That's what we're trying to not let happen. Details. We got to be prepared. So if we're going to close the herd for 120 days, where the heck are the gilts? If they're not on site, are we going to be able to expose them somewhere else? What if you got one gilt source feeding 5 sow herds? What are you going to do when you break one sow herd? There's a lot to think about, but it's easier to think about it ahead of time than when the fires are burning. Timing's critical. We've got to be there early. We've got to capture enough virus. We have to get it while it's still being shed, and it takes a lot of virus. We want to keep the virus so that we're now in a different world. We're trying to make the virus live so it helps us. So we're going to handle it cold because it really doesn't die out much in the cold, but as you can see, heat kills it. We've got to have prompt and timely euthanasia for debilitated chronically infected pigs. You saw the pickup. Do you have a euthanasia plan? This is my pitch for CO2 chamber, something else, because that's an awful lot of debilitated pigs to have to euthanize. If you don't have a plan, it's a good time to think about it. Then, before you leave, after we've done all this feedback, we got to destroy and disinfect all materials because we don't want to track it to the next place. Here's kind of what it looks like in the outbreak so far. Nobody lives the first week, nobody lives the second week, about 1/4 of them the third week, but we got to get them out of there because we want a clean, disinfected, empty farrowing. 4th week, 70%. By the 5th week, because sow immunities come rolling up, pigs are alive. And now you're back to weaning normal-looking pigs, but they're going to scour in the nursery. There's going to be some hit in there. So what are the losses and the impacts going to be? Individual sow farm, just to do the math, to get the perspective, going to lose about 5 weeks worth of wean production, pig production out of a year. We're going to have 4 weeks of acute pig death loss, but everybody forgets that all of those sows that were sicker than the devil that first week or so, 20 weeks later, you're not going to have very many pigs when you try to breed them back. So you just as well throw that week out too. They're gone. So there's 5 weeks out of there. Growing pigs are going to lose a week of growth. This is for Scott. I needed to get a finance one in here, Scott, so that it was really understandable. So for the finance people in the room, all you have to figure out is how to cover 52 weeks of expenses with 47 weeks worth of revenue. That's what we're going to give you. Real simple math. For the industry, this remains to be seen. This is going to be interesting right off the bat. There's going to be a regional impact on slaughter pig supply because realize that the pigs that aren't there because of the outbreaks in May just should be going to market after Thanksgiving. We haven't even seen this in the marketplace. So what happens to the Guyman plant? They were counting on all those pigs, weren't they? Well, now what about Iowa? What about the holes in there? What does it do to the markets for wean pigs? Did a vet-to-vet last week. Proved the sale of a bunch of pigs, $86 a wean pig. 3 days later, it's 106. And so it's going to have some real spot market impact. Slaughter weights, this is a big deal. Okay, so your herd breaks. What's the very first thing we're going to think of? I know John knows the answer, that's why I look over there. The first thing I'm going to do is I know I'm going to have some empty finishing barn. I'm going to put 10 more pounds on every pig as quick as I can. I got to make up that money somewhere, and I'm going to have some space. So slaughter weights are going to have to go up to fill the production. What's it going to do to cost of production? I'll bet we know next year at Swine Day because we're going to wait. I want to see your feeding trials on PED recovered pigs in the nursery and how fast they don't grow. It's going to have an impact. So, we've done all this PED.
Since you mentioned next year at Swine Day.
Yeah.
All right. So remember the graph I showed where the cases are going up and up and up? Those are the ones that are going to hit us.
Yeah.
And that impact is going to be real dramatic.
So when I step away from that, I've got to say, okay, but as a veterinarian, guess what? PERS is still here. So I'm out here playing fire engine fix-it-all. I could just as easily be moving PERS virus around in the middle of an epidemic and make life worse on another side. And you remember the $440,000? PERS still costs this industry $1 billion a year. That's why we don't want to waste a good crisis, because it's time to reset the dial. And the good news is that every biosecurity improvement we make against PED is 2 for 1. Counts double for PERS. So we change our behavior in how we move pigs around. We're going to change PERS and PED and probably salmonella and a few other things. The other good news is the industry understands what we're going to do. We don't have to start over and say, well, this is what we're going to do in this situation. And it's going to radically rework this sanitation chain from the farm to the plant. Bad news, we're going to end up with a bunch of endemics, regardless how hard we try. It's already pretty obvious from the cases that broke early. Some of those recovered, cleaned up, negative pigs. Others are now having sow herds going back through rebreaks, same sow herd. So 6, 8 months later, here they go again. That's endemic. That's really, really painful. It's really hard to go in and turn an endemic herd and make it acute again and clean it up. All the veterinarians, for all the confidence we may have standing up in front of an audience talking about how much we think we know, we don't have a validated procedure or method. We're just living on our TGE experience and hope to heck it works. We don't know. So anybody that says they got it all figured out, that's good.
And I think the other thing I would add is the good old TGE days, a lot of the younger veterinarians have never really seen good old TGE the way it used to be.
Yeah. And just as an old guy, the boots on the ground, we're kind of short on putting enough veterinarians in the hot spots at once to do the job. We're gaining, but this isn't a lot of fun out there, you might guess. So with that, Dr. Hesse and I would welcome questions, comments, sympathy.
Questions?
Yes. The question was it here before May? And the answer to that is yes. Iowa State had 500 samples that they had in their collection, and they retrospectively looked back all the way into December. The first documented case that they could detect came out of Ohio, actually, and that was about 3 weeks prior to the first ones that were showing up in Iowa and Indiana. They could not find any other cases prior to the middle of May. And I don't know of any other lab that has.
Dr. Odie.
My question is, you alluded several times to field evidence of aerosol transmission, but you never told us what that evidence actually is.
Field evidence of aerosol transmission. What was the evidence? Dr. Luke Dufresne, seaboard veterinarian, excellent veterinarian in Kansas City. When their farms broke, some of the patterns of the way they broke with no traffic between them suggested they could find no other reason for it to move the distance it did. That said, well, I mean, the wind moves kind of fast out there, but you could move particulate manure quite a ways. This is not quite the same as what we're talking about with influenza or mycoplasma. Right. This could actually be moving material.
That was back in what, July or so? Recently, May, July, yeah, June. So recently, we're seeing similar patterns from, remember 2-3 weeks ago when we had the really strong south wind? Well, about 3 days after that, a lot of farms started breaking.
But there's also a lot of manure on the ground right now, so it's getting colder. Yes, Doug.
On your challenge, is there indication that different isolates have different failure rates?
At this point in time, in the U.S., we believe there's really only one PED. Several labs have sequenced the viruses that they have, and all the sequence is coming back virtually identical. Will that change over time? Sure it will. These viruses, the only constant is change. But at the moment, we believe it's just that Chinese virus.
Other questions?
That's it. Dan Garrity.
From a reproductive standpoint, strictly not PERS included, is there any real reproductive problems in the sow farms?
Reproductive problems in the sow farms post the acute phase. Not like PERS at all. The ones that are because the sows get sick when it's first there. It's the sows that didn't eat for 4 or 5 days in that first week that are a problem, but no direct reproductive impact, they come roaring back.
One thing that is of concern, and that's why Steve emphasized what else is there in the feedback material, because you don't want to make a bad situation worse, is some of the other things that come along with that feedback. And there are some reports where they did not return to what the normal production level was. And it's probably not the PED, but it's the other little friends that came along with it.
Okay, well, let's thank Dick and Steve. Thank goodness we have them.